Assalamu alaikum. I am Dr. Haiba Kaul, working as Associate Professor of Genetics at University of Veterinary and Animal Sciences. Today, I will present how whole exome sequencing identified a novel mutation in the UCY2D gene in a large consanguineous Pakistani family that diagnosed it to Leber congenital amaurosis 1 LCA. LCA or Leber congenital amaurosis is a rare inherited eye disorder that affects children mostly at birth or in early infancy. It basically it affects the cells known as photoreceptor cells that are present at the back layer, very thin layer that is known as retina. And these photoreceptor cells are known as rods and cones. Cones control the color vision and the central vision, and it is responsible for providing vision in during light time, while as the rods they affect the peripheral vision and the night vision. So any disruption in these rods or cones can cause visual abnormalities in children, and it can also count for childhood blindness. Key features of LC include nystagmus, photophobia, hypermetropia that is known as far sightedness, allied visual abnormalities such as glaucoma, keratoconus, cataract, they are also associated with it. LCA is mostly is inherited as autosomal recessive disorder. It's a Mendelian disorder, mostly monogenic, and about more than 29 genes they are implicated in LCA. Its various types are known according to the mutation is responsible for the disease. So DUCY2D gene is responsible for almost 12% of the cases of the LCA, and it's also known as LCA1. If you look at this picture, you can see here that so LCA1 in LCA1 GUCY2 mutations, it encodes this gene encodes an enzyme known as ROS-GC1. It's an enzyme that is important for this phototransduction of rods and cones, and it converts GTP to cyclic GMP. And cyclic GMP is essential for the regeneration of the phototransduction channel. So, due to if there are any mutation, most of the mutations in this gene are loss of function mutations. If any mutation occurs in this gene, this phototransduction channel is disrupted, and as a result, vision loss can result. So, this is the consequence of the GUCUY2D mutations because it's autosomal recessive. So, most of the result of the mutations that results in the loss of function or the reduction in the enzyme activity. So normal enzyme, it helps in the restoration of cyclic GMP, whereas the mutated enzyme disrupts cyclic GMP and the disruption of the photoreceptor function, and it can cause retinal degeneration. It's a progressive disease. So as the child progresses, vision problems, they are also increased. So why molecular testing or genetic testing is essential or important for this type of disorder? It's a very clinically heterogeneous disorder. So there are several clinical features, and different patients can have a different set of clinical symptoms. The family that I identified and designated as BL01, that family was not diagnosed through clinical methods or even through the candidate gene approach. So, genetic testing, it enables genetic counseling, it helps in the diagnosis, and it guides patients how to deal with prognosis, where this gene is involved. And the diagnostic tools nowadays in various tools of NGS, they are there and they can help to identify the cause of the disease in different patients. So we identified this family from Lahore. It's a large family, and we collected blood samples from these 7 affected individuals. They were brother and sisters, and 6 unaffected members. There were several other unaffected people in the family as well, but we couldn't get their samples. After sampling was done after informed consent, and after recruitment, clinical evaluation was done. Blood samples collection was done, DNA extraction, and then whole exome sequencing was performed on a proband. And when a variant was identified through WS, we performed Sanger sequencing to all identified individuals to check the segregation status, and then this the identified variant that was classified for its pathogenicity according to ACMG criteria. So, clinical and comprehensive clinical analysis was done using visual acuity, pupillary response, and several by the trained ophthalmologists. Other allied features such as glaucoma, cataract, photophobia, nystagmus, they were also characterized and checked in different patients. ERG was also done, but it was a non-responsive in almost all the patients. Blood samples were collected, DNA was extracted, and with the help of our collaborators at 3 billion in South Korea, we performed the whole exome sequencing using Illumina Nova Seq 6000 platform. Variant filtering and prioritization was done using several bioinformatic filters. Population frequency was set as less than 0.1%. Homozygosity mapping was done. Several bioinformatic tools like SIFT for polyphon CAD, mutation tester, they were used to identify the and classify the pathogenic variant. Lastly, we did Sanger sequencing in all the affected members, and then the segregation analysis was done, and then we classify the pathogenic variants using several bioinformatics tools. So these patients show classical symptoms of LCM. This family, as I told you before, we couldn't classify this family, diagnose this family using traditional clinical methods and genetic analysis. So after WS, after whole exome sequencing, we were able to diagnose this family, and severe photophobia was a hallmark of this disease in all the members. Some patients had cataract, some had glaucoma, and others there were some were present. Pendular nystagmus was present. However, ERG was unrecordable in all the affected individuals. So, whole exome sequencing identified a novel mutation in exon 2, C315 cytosine to adenine change, and it results in the amino acid change at 1 of 5 position. Cysteine was replaced by a premature stop codon in exon 2. So, as it was a recessive disease, so because of this change, a stop codon is formed quite early in the transcript, and it undergoes non-sense mediated mRNA decay that was performed using SETA and bioinformatics tools. So, likely effect, although it was all done through bioinformatics, so it was not a functional study, reduced enzyme activity and disrupted cyclic GMP production can be its effect, and it's also reported in certain other studies where they performed some functional analysis. Here you can see these are the bioinformatic predictors of the pathogenicity of this identified variant. You can see WebCAD mutation tester. They all say it's likely pathogenic and high confidence in pathogenicity of this variant. And these are the certain domains. As this change was present in the amino acid 105, you can see only signal peptide domain was retained, whereas the extracellular domain was partially lost, and all the other domains they were lost due to this mutation in BL01. This is the pedigree. You can see here these are the 7 affected individuals that participated in this study, and all of these were homozygous for the adenine AA, you can see, and these are the heterozygous individuals, and this is the only one we found that was the homozygous for the wild type allele. These are the parents. These are the results of sequencing. This is a sequence chromatogram. This is the heterozygous carrier, and you can see this is here. This is a homozygous affected individual with homozygous adenine, and this is the heterozygous. This is the ACMG classification of the variant, and you can see all its 5 criteria. This pathogenic mutation is very strongly to moderately pathogenic, where this variant, nonsense variant, results in the loss of function mutation. So it was absent in population databases, that is consistent with rare disease causing mutations. And although this stop codon is present in the very early in the transcript, so effect where a nonsense mediated decay is predicted. So the conclusion of this study is that GUCY2D gene was considered as the pathogenic mutation in this family, which resulted in this change from cytosine into and that converted to stop codon, and that can explain the severe phenotype in this family. So the ACMG criteria also supported this interpretation of this way. Thank you very much for your attention.